I think in phosphorus world, phosphate gets all of the attention for good reason, because that's generally the form in which we find phosphorus with its 4 oxygen companions. But we're going to find out today a little bit more about another form of phosphorus, phosphite, and its promise for addressing issues of pea sustainability and plant production. So Dr. Luis Herrera from CINVESTAV is, please welcome him to the forum.
Good morning, everyone. I'm Luis Herrera Estrella from Irapuato, Mexico. I'm director of the National Laboratory of Genomics for Biodiversity from the Center of Research and Advanced Studies in Mexico, and co-founder of a little company who wants to commercialize an interesting technology. So I've been working for the past 20 years on phosphate uptake and phosphate utilization efficiency. And the main problem we have in agriculture is from what you apply into the soil, 70 to 80% gets fixed into the soil. And the crops use only 20 to 30%. How much it is lost to soil fixation depends a lot on the soil characteristics, on the soil chemistry. Acidic and alkaline soils fix more readily phosphorus than neutral soils. So there is also a loss because of microbial activity. Microbes also want to utilize phosphate, and they convert it into organic forms that plants cannot readily uptake. So that is a major challenge, and that's why we are using a very high level of phosphate fertilizer. So I've been working on different strategies how to approach this problem. I have been working on the role of root architecture in phosphate uptake and different metabolic processes to enhance phosphate utilization. But unfortunately, not only my group, but most of the groups working on molecular biology and genomics of these processes, we have not really achieved any major advance compared with traditional breeding. This is a very quantitative trait which requires many components, and we have not really found a magic bullet to really have a significant advance. So we wanted to change the concept and tried something completely new. If the problem is the chemical properties of phosphate, why don't we use a different phosphorus molecule, which doesn't get fixed into the soil? The problem of phosphorus soil fixation is illustrated in this slide. All the red and yellowish areas have big phosphorus fixation problems. As you can see, most of the world has problems except some areas in the United States and some areas in Northern Africa. But most of Europe, most of South America, Mexico, Australia, China, India, the Soviet Union, we all have problems of high phosphate fixation. And the problem is that phosphate gets very readily fixed into the soil because it reacts with cations in the soil. In acidic soils, it reacts with iron, aluminum, and manganese, and in alkaline soils, it reacts with calcium. So it forms insoluble compounds that the plant cannot take up anymore. And it's only relatively available for plant to take at neutral pHs, but it's never 100%. So even at neutral pHs, we have only a relative availability. So we searched for different compounds that could be used as alternative sources of phosphorus to fertilize plants. And we bump into phosphite. And it's quite interesting because we found a paper from 1945 from some German chemist suggesting that phosphite was a superior component to fertilize plants because of its physicochemical properties. Phosphite solubility is less dependent on pH, and phosphite is less reactive than phosphate with soil components. So it is about 1,000 times more soluble than phosphate. And we also found some interesting things. Phosphite is already widely used in agriculture and as an effective treatment against oomycetes. It is actually not a fungicide, but it is sold either as a fungicide or as a fertilizer. Companies who sell phosphite as a fertilizer do it so because they want to avoid the FDA. So if you want to sell a fungicide, you have to go through regulation of FDA. If you want to sell a fertilizer, you don't go through FDA, and you just say that the benefit of the molecule is because you are fertilizing the plant, but the effect is actually on fungal infections. And there is no toxicity reported for humans and animals. It's been already approved for use in agriculture, is already approved for use in food industry. In Europe, in US, Canada, Mexico, many places. The problem is that plants cannot use phosphite as a phosphorus source. So this is corn plants without fertilization, just living on the natural phosphate that is present in the soil. If you fertilize, the plants grow better because you are adding enough phosphorus to fully support the development of the plant. But if you add phosphite, then the plant grows less than without phosphate. And the reason is simple. Phosphate and phosphite enter into the plant through the same gate. So if you have a crowd of people, of which 80% are phosphite molecules and 20% are phosphate molecules, you will get more phosphite than phosphate, so you will get a reduced entry of phosphorus into the plant. So it's a very simple concept. And this has been shown to be the case for many crops, beans, lettuce, soybeans. So no plant to date has been found that can utilize phosphite as a phosphorus source. So this is under field conditions. If you apply phosphite, the plant doesn't grow. If you will apply phosphate, it grows. So given the advantage of phosphite as a potential fertilizer, can we engineer plants to metabolize phosphite? And the answer is yes. We found that some bacterial isolates are capable of using phosphite as the sole phosphorus source. The reaction is very simple. The bacteria takes up phosphate, it reduces it to phosphite, and it oxidizes into phosphite and reduces NAD to NADH. So you start with a molecule which is not toxic, and the products are two molecules that are present in all living cells. The enzyme is very highly specific. It can be used only phosphite, not nitrate, formate, glycerate, arsenite. It's very specific, so you don't expect it to have other activities. And then the question is, what happens if you introduce this gene into plants, express it, and see how they behave in the presence of phosphite? So this is a control plant. These are tobacco plants with no fertilization, so it grows very little. This is an artificial substrate which doesn't fix phosphorus. If you add phosphate, the plant grows very well. If you add phosphite, the plant grows less well than without fertilization. If you introduce this gene and express these genes in the plant, then the plant grows in phosphate, and it grows now in phosphite. We haven't found any morphological or biochemical difference of the plant grown on phosphate or phosphite. They seem to grow identically at the same rate. The interesting question is that if phosphite indeed doesn't fix into the soil and phosphate fixes into the soil, when you use an agricultural soil, you should be able to produce the same biomass with less phosphorus using phosphite than phosphate. So these are some experiments in which you can see that the transgenic plant with phosphate grows less well than with phosphite. So the control grows the same than the transgenic with phosphate, but the control doesn't grow with phosphite. So the interesting is when you do the numbers, if you take plant biomass or seed biomass or whatever parameters you can use, then you have the transgenic and the control, and you see that there is no difference in total biomass production or seed biomass production. In fact, they are identical. But what happens is when you fertilize with phosphite, if you see here, there are different fertilization regimes, 10 ppm, 30 ppm, and 60 ppm of phosphate, and 10, 30, and 60 of phosphite. So when you add 30 ppm of phosphite, you have the same biomass as you do with 60 ppm of phosphate. So you can reduce 50% of the amount of fertilizer of phosphate fertilizer. These plants were fertilized with everything normal, except that phosphate was replaced with phosphite in some pots. And this is an agricultural soil in which we didn't prevent any leaching because we use potassium phosphite. There is no formulation, it's just the compound mixed into the soil and then water with the nutrient solution. So leaching and everything was allowed. So even though you expect to have a lot more leaching from phosphite because it's more soluble, you can still save 50% because the plant is more efficient in taking up this molecule, which is always 100% available. Properly formulated, we expect that we can reduce the amount of phosphate fertilizer you apply to 30% or 20% of what you need. The other interesting aspect of this technology is weed control. What we have done is tailor crops to establish a selective fertilization strategy. So you use phosphite that will be used only by your favorite crop, but no other plant can use it. The problem with weeds is that because of the abuse of the technology, herbicide-resistant biotabs have exploded in the past 2 decades. So now, cotton growers in the US are facing the problem that they cannot use Roundup Ready seeds anymore because the most aggressive weeds have become resistant to Roundup. Monsanto came with a second herbicide resistance, but this herbicide is volatile. So the problem is that you treat your plants, they are resistant to the herbicide, but your neighbor that is not using the resistant plant, the herbicide travels to the other field and then kills the crops of your neighbor. Thing that didn't happen with glyphosate. So the question is, can we do innovative weeding without the use of herbicides? So the concept is simple. Phosphite is not a herbicide. So phosphite doesn't kill plants. Simply, plants cannot use it as a phosphorus source, and if you feed them, you reduce their growth because phosphite is competing with an essential nutrient to enter the plant. This is a simple field experiment just to test it, to test this hypothesis. What happens if you treat a natural agricultural soil with phosphite? So this is a non-treated plot, and this is a treated plot. So you can see that there is a strong delay in the emergence of weeds in this field. So it works. All natural weeds present in this soil is most of these experiments we did in Argentina because in Mexico we are not allowed to do many of these things. So it works nice. This is a comparison between glyphosate treated, phosphite treated, and non-treated. So you can see the weeds growing here quite strongly. Glyphosate kills everything, and that's what the farmer wants to see now, that you spray once and you kill everything, even your neighbor, if possible. And phosphite delays very much the emergence of the weeds, but it doesn't kill them. So this is a field experiment with tobacco plants. Non-treated tobacco plants were here, but weeds grew faster and killed the plants. Treated with 180 ppm of phosphite, which is the recommended dose for these fields in Argentina. And you can see here now the tobacco plants dominating the system, although the weeds are still here. These are tobacco control plants. You can see here they are small, and these are the transgenic. You can reduce down to 80 ppm, and you have the same result, weed control and promoting your crop growth. The system can be applied to many crops. This is corn. This is an experiment with a segregating population, so you can see we don't cheat. So plants are mixed in the same pot. Null segregants and transgenic. Null segregant, transgenic. Null segregant, transgenic. It can be used also for soybean, control, transgenic. It has been shown to work also for rice. We are testing it for sugarcane, but we have done a lot of work with cotton, which I will describe to you quickly. One of the interesting things about the system is that the gene itself can be used as a selectable marker. So you don't need to add a herbicide resistance or an antibiotic resistant genes, because you can directly select for regenerated plants that are able to metabolize phosphite. So these are just control plants in tissue culture. This is soybean grown on phosphite, nothing grows. If you transform with the gene, then you get regenerated plants at a very high efficiency that are transgenic. So these are some experiments that we did, competition experiments with different weeds. This is a broad leaf weed, and you can see that the weed is completely dominating the system. This is a climbing weed. This is the cotton, and this is the weed with phosphate. If you apply phosphite instead of phosphate, then you have your cotton, and your weed is here. So you are not killing the weed, because most of the agroecology says that killing weeds is a crime, so we are not killing weeds. Weeds are there, we are just reducing their growth to a little bit less of what they normally grow on a low phosphate soil. This is with a grass type of weed, than the cotton. If you clean up with phosphite, then the plant outcompetes the grass, which is still there. And you can see here the effect in biomass production. The weed competes with your crop with no phosphate. And the interesting thing is that when you fertilize with phosphate, the weed completely takes over the system. When you have little phosphate, they sort of share life. But when you fertilize, the weed completely outcompetes your crop, and then you need a herbicide to kill the weed. But if you apply phosphite, you reduce the growth of the weed and enormously promote the growth of your crop. So the system works quite nicely. So we did some experiments using agricultural soils with their natural weeds. So there were about 15 different types of weeds in this soil. You can see here how they grow. You can hardly see the cotton here. You have always a mixture of cotton plants growing with the weeds that we intentionally planted in the pot, but these are natural weeds. This is really aggressive. When you treat with phosphite, your cotton plant dominates the 20 different weed species. We have even tested the glyphosate-resistant Palmer amaranth, which is one of the most noxious weeds, and it is effectively controlled by it. So one of the interesting things is that this is a close-up of the system. You don't kill the weeds, so weeds allow you to prevent water evaporation and soil erosion. Because the problem is that when you use a herbicide, everything is empty here. So soil erosion is a major problem. And this is just a comparison of the tobacco plants grown with 50% fertilizer, 10% herbicide, versus 100% PI fertilizer and 100% fertilizer. So it grows quite well. It has many advantages. If phosphite is about 60% more expensive, currently, phosphite is about 60% more expensive than phosphate. If you reduce 60% the amount you need to grow your crop, you break even. But then you save all your money on herbicides. But that's not the most important thing. The most important thing is the environmental impact. An excessive use of phosphorus causes water eutrophication, hypoxia, and is a huge problem that we share with the United States. In the delta of the Mississippi River, there is the largest ocean dead zone in the world. It's 47,000 square kilometers that we share in the Mexican Gulf with the United States, and which has not been reduced by 1 kilometer in the past 10 years, but it has exponentially grown. So we need to reduce the amount of phosphate we apply. We started testing whether microalgae and algae, which are the cause of algae blooms, which cause hypoxia and produce toxins and kill off the marine life, could use phosphite as a phosphorus source. They are not able, this is Chlamydomonas reinhardti, with phosphate and with phosphite. So they don't grow. So even if phosphate is released into the ocean, it will not cause algae blooms. It will slowly oxidize into phosphate due to the presence of oxygen, because oxygen naturally oxidizes phosphite. But you can also produce transgenic microalgae that can use phosphite as a phosphorus source. I'm going to go faster. So one of the problems to use microalgae systems, this is a wonderful system because you don't need to feed sugar or anything to your microalgae. So you use sunlight and CO2, and then you produce your microalgae with your favorite compound, you name it, omega-6, omega-9, pigments, oils, whatever. The problem is that you cannot use open systems, which is the dream of everybody. You need to use closed reactors that you have to illuminate, and that is very expensive. But what happens if you modify your algae to metabolize phosphite? So we have done similar competition experiments to what I show you with plants, with the algae versus an engineered Chlamydomonas. Similar results. But then we bump into an expert in these topics, and he says, you have to do a real world experiment. Go and take water from a natural source and makes this mix to compete with your favorite algae, with your engineered algae. So we went to a canal. This is an agricultural region. You can see, well, you cannot see here well, but it's very green. So it has many algae. So what happens if you take your Chlamydomonas capable of metabolizing phosphite, but you fertilize the system with phosphate? So initially, you have a very high inoculum of your Chlamydomona, so it dominates the system, but by day 6, everything is growing but your Chlamydomonas is dead. What happens if you fertilize with phosphite? Then your Chlamydomonas is growing here, and your Chlamydomonas is growing here. So it's capable of controlling all the algae and other bugs that are growing in this water. So this system can be used also for the industrial production of biocompounds using microalgae as bioreactors. So we have done it. These are 100-liter reactors. They are open here to the open air. You can see the same strain growing with phosphite and phosphate. This reactor collapses because of contamination. This doesn't collapse because phosphite is able to control contamination and promote the growth of your algae. Just to end up this Our technology was corroborated by a group in MIT, which six years after we published our first paper, they sort of independently discovered the same technology and applied it to fungi. And this is the people who did the work. Damar Lopez Arredondo did all the initial work on the phosphite used in plants and algae. Thank you.
Okay, I think there might be some questions and discussion of these exciting data.
So how many years can the phosphite fertilizer actually inhibit the weeds, like in the long run?
What do you mean?
So like long term, those.
If weeds can evolve.
No, not the evolution thing. It's the oxidation thing.
Okay. So the idea is to apply phosphite fertilizer, formulated in the right way and in the right dose, so that the crop uses 95% of it. The remaining 5% is going to be oxidized within 4 to 6 months to phosphate by the presence of oxygen in the soil. So at the end of your cycle, there will be no phosphite left. So if you want to rotate with a non-engineered crop, you can rotate.
Yes, so I think you got to my question in the last slide, but it kind of passed by me a little bit. How do you produce the phosphite? Is that a microbial process?
No, it's a chemical reduction process. There are a number of companies that produce phosphite. Probably in the worldwide scale, maybe 500,000 tons of phosphite are produced per year. There is a cheap source of phosphite, which we didn't know about. Many electrochemical plating industries use hypophosphite as a reducing agent to cover with metals many things. And they produce highly pure phosphite. So we got an offer from a company who said, well, we have 10,000 tons per year of phosphite. If you want them, you can take them for free. So, but of course, it's nothing compared to the 30 million tons we need. So eventually what we need is to produce more phosphite. That comes from phosphoric rock. But the idea is that if you use 100% phosphate, you reduce to 50, 40, or 30, or 20%. So it will double, triple, or quadruple the lifetime of the reserves. And it's going to be the only technology that can be used in Mars or the moon, if we ever get there, because of the phosphorus present in those, because there is no oxygen, it's all in as phosphite.
Okay, we're going to take one more question. I think it's appropriate that he gets to ask that one.
With 1,000 times more solubility in soil, what's the relative loss rate of phosphite versus applied phosphate in the surface runoff and leaching and drainage?
Yeah, it has a higher rate of leaching. That's why we need to formulate it in the right way, so it's a slow release fertilizer. We have already formulations which make the plant, even with normal irrigation, to use about 90% of what we apply.
Okay, great. So I know that many people have more to ask and discuss with Luis during the rest of the day. That was a very eye-opening presentation, I think, for many of us. So we're going to move on to the next part of our. Oh, let's give Luis a big hand. Great idea.